Welcome to this module on plant containment. We will introduce you to the key concepts associated with plant containment. This module is part of IB6043, the course on plant molecular biology. Plant containment is required because of modern biotechnology, modern breeding methods, the internationalization of trade, the evolution of disease-resistant pathogens, the development of biological control agents, and the need to comply with national laws and international laws have all contributed to the development of plant containment methods. The objective of this module is to introduce you to the concepts of plant containment, to identify relevant situations which require plant containment, to become aware of the national laws, policies, and guidelines pertaining to containment, to give an insight into the international guidelines, to provide information on the design of plant facilities in vitro, and the design of transgenic plant containment facilities. Upon completion of this module, you should be able to describe situations in which plant transgenic containment is needed, describe the relevant national laws related to the development of living modified organisms, demonstrate the ability to access and to understand international and national policies and guidelines, and to develop a conceptual design for a plant transgenic containment facility. Under what situations do we need to resort to containment? The goal of biosafety with regard to transgenic plants is to prevent dissemination of the modified species outside the growing area. Containment is crucial in transgenic plant research in the labs, greenhouses, and growth chambers. Field trials with transgenic plants require compliance with Malaysian biosafety regulation. Containment is primary and secondary. So this symbol represents the biohazard. In this case, it may represent the plant which is genetically modified. The first barrier is known as primary containment, and this barrier protects the laboratory worker from the biological agent or biohazard. The second level of containment protects the environment from the biological agent. Both primary and secondary containment are necessary in order to protect the laboratory worker as well as the environment from the biological agent. To summarize, the purpose of primary containment is to protect the laboratory users from the biological agent. The purpose of secondary containment is to protect the environment from the biological agent. The following situations will require containment. First, experiments involving genetically modified plants, imported germplasm which may contain pathogens of exotic origin, plants which have been cultured in vitro, challenge tests involving pathogens and pests, and controlled breeding experiments. The following risks are posed by genetically modified plants. First, we have transgene escape via the dissemination of pollen. Genetically modified plants can have impact on pollinators. They represent potential bioterrorism and bioeconomic threats. And economic impacts can result from the release of germplasm which is genetically modified. Quarantine of imported planting material, including biofertilizers containing living organisms, is mandated by national laws. They pose a possible risk, which include the introduction of exotic invasive species and loss of productivity in native land races as a result of exotic pollen. Plants developed in vitro may need containment due to the fact that they have not adapted to agronomic pests which are present in the ecosystem. Finally, controlled breeding experiments may have to be carried out under containment in order to curtail the genesis of indeterminate F1 hybrids. Indeterminate hybrids can lead to a major loss of revenue to crop breeders. Challenge tests involve the use of biological agents, including microbes and insects, which can cause damage to plants if released in the environment. These situations require containment. The first section of this module has focused on the basic concepts of containment. The next section will focus on the guidelines for containment. Thank you for watching, and the next module will follow.